Hi all, Dr. Clark here again, and today we're going to switch gears a little bit. We talked about how carbohydrates are constructed through photosynthesis, and now we're going to kind of talk about how carbohydrates are destructed through cellular respiration. So how do organisms like ourselves and all other organisms that have mitochondria, how do they break down sugars and fats and proteins and nucleic acids to get energy? Well, the first process occurs in all organisms, regardless of whether or not they have mitochondria, and that process is glycolysis. So here you can see just the initial steps of glycolysis, and we'll go into much greater detail with this as we progress, but just to give you kind of an overview of what we're going to talk about today, we're going to talk about glycolysis, and that's the process of taking a glucose molecule like this. So remember, glucose is C6H12O6. So this is showing the 6 carbons. We have to supply a little bit of ATP, which we call the energy investment phase. In doing that, we phosphorylate the carbon molecules, and so this molecule gets split in half. And so you get 3 carbons on each side with a phosphate, and this is called glyceraldehyde 3-phosphate. And you might remember that from photosynthesis when we were talking about glyceraldehyde forming a glucose molecule. When we deconstruct a glucose molecule, similar process. That glyceraldehyde 3-phosphate then gets further processed, right? And we call that being oxidated because we're going to remove electrons and hydrogen ions from it. And then we're going to utilize the energy to reform ATP. And so we're going to have an energy investment phase where we're supplying ATP and an energy payoff phase where we're obtaining ATP. So we're going to look at these a little bit more in detail and go through all the steps to glycolysis and talk about kind of what organisms have and the process by which this can occur. All right, so often we're interested in, well, at least initially we're interested in, where does the energy come from in food? So we know that we have to consume food to get energy. All organisms have to consume food to get energy. And that's one of the 5 characteristics that make an organism alive. Remember, we said that an organism must have a metabolism. So that means it must be able to convert some type of food into energy. So where does it come from? Well, it comes from the movement of electrons, the borrowing of electrons from one molecule to supply that to another molecule. And that movement of those electrons is the energy that we're talking about that allows us to drive work and do things like run our nervous system, fire our muscles, allows for us to have muscles to talk with, digestion muscles, allows for us to thermoregulate our body at 98.6 degrees. It allows for other organisms to do whatever they do, swim, fly, whatever it might be. It's the passing of electrons from one atom to another, from one molecule to another, that gives that organism energy. Now we have special terms to deal with that movement of electrons. And so anytime an atom or a molecule loses an electron, it's considered oxidized, or it goes through an oxidation reaction. Anytime a molecule or atom gains an electron, it's considered reduced, or goes through a reduction reaction. Now, I know that most people, especially the first time you hear this, you're like, reduction? You sure you don't have those backwards? Okay, reduction in this case is often coupled with a reduction in charge. So if it did have a charge, By gaining that electron, it now becomes neutral. That's not always the case, but reduction events always gain electrons. And these are called redox reactions or oxidation reduction reactions. And they always occur together. So, we can kind of go back in time a little bit to where I was talking about energy. And when we were talking in chapter 5 or, lecture, I don't know, maybe 16, somewhere in that range, maybe 15, where we were talking about endothermic or exothermic and endothermic reactions. And we were talking about activation energy and whatnot. And I said endothermic reactions need energy supplied in order to form bonds, and the product is worth more energy than the substrate. And exothermic is releasing energy. The product is worth less energy than the starting point. You can think of that in the exact same form as oxidation reduction. Right? Oxidation loses an electron, so this is much like an exothermic reaction. And indeed, in many cases, they are the same. Right? A reduction gains an electron. This is much like an endothermic reaction. It gains energy. And remember when I said endothermic reactions always occur with exothermic reactions. So the exothermic reaction is going to supply the energy for the endothermic reaction. The exact same thing is true here. Oxidation is always going to occur with reduction. And when that happens, right, the loss of an electron, so if a molecule is losing an electron, right, it is going to have less energy or a lower energy than the molecule that gains an electron. So here you can see this molecule lost an electron. Okay. Therefore, now it's a proton. It has a positive charge. Okay. So it's now a polar molecule. And this one here gained an electron, and now it's neutral. Okay. Now this is not always the case. It won't always be neutral, but it will always, if it's gaining an electron, it'll always have a higher energy level than the previous molecule, than what it was without that gain of an electron. Okay? So hopefully that makes sense. And we're going to go through quite a few examples of this, so it should make sense as we progress through. Okay. So to further look at energy and to further examine this, we need to talk about how organisms go about harvesting energy from the carbohydrates that were constructed in photosynthesis. Okay, we just got done talking about how organisms, plants, algae, photosynthetic bacteria, some photosynthetic protista, how they go about constructing carbohydrates. Now we're going to talk about how those molecules get broken down. Right? Now the process is nearly the exact opposite of photosynthesis. So if you remember the photosynthesis equation, where you're going to supply carbon dioxide, you're going to supply water, you're going to supply photons, and that's going to yield carbohydrates and oxygen and a little leftover water. Well, the process of cellular respiration is nearly the exact opposite. We're going to supply glucose, that carbohydrate. Okay. We're going to supply oxygen to the equation. Okay. That's going to yield carbon dioxide as a waste product, water as a waste product, metabolic water, although I shouldn't call it a waste product. It's not the product that we're after, but it's useful, it's usable. Carbon dioxide is poisonous, so that's a true waste product. And then the final piece, energy. And that energy is going to be in the form of ATP, but often it's also in the form of heat. So it's lost to the organism. So some of the energy from breaking down that glucose molecule is going to be lost to the environment or utilized to maintain a constant body temperature for some organisms. And for others, it's just going to be converted into ATP. So cellular respiration in general takes place in two main stages. One, glycolysis, and that's what we're going to talk about mainly here. And the other one is the Krebs cycle. Now, glycolysis occurs in the cytoplasm of the cell, or the cytosol of the cell. And no oxygen is required for glycolysis to occur. It generates ATP from the breaking down of a solid glucose molecule. The process of glycolysis, it'll break that glucose molecule into pyruvate. Pyruvate then enters the Krebs cycle, or a precursor to the Krebs cycle, which we'll get to. The Krebs cycle occurs in the mitochondria. So only organisms with mitochondria can go through this cycle. And so that Krebs cycle is going to then further break down the products of glycolysis, pyruvate, and that pyruvate is going to be processed into a molecule which we call acetyl-CoA. It's going to further break that down and get more ATP and also get more electrons, so we can utilize those electrons in the electron transport chain in order to produce ATP. We're going to come back to this piece next time. Today we're just going to talk about glycolysis. Okay, so this is an overview of what the slide before was showing. And remember, we're just going to be concentrating on glycolysis, but I'm going to give you kind of an overview of all the steps, just because it can get complex a little bit, and the more times you hear complex things, the easier it will be for you to learn that information. So glucose molecule is going to come in. Glycolysis, there's just a bunch of enzymes that work in glycolysis to convert that glucose molecule into ATP, remove an electron, hydrogen, to form NADH, and create pyruvate, 2 molecules of pyruvate. Pyruvate then enters the mitochondria, then goes through an oxidation phase, which we call pyruvate processing, or there's an enzyme called pyruvate dehydrogenase that changes the molecule, kicks out a carbon as carbon dioxide, adds an electron to NAD to form NADH, and acetyl-CoA is formed, 2 molecules of that. Acetyl-CoA then enters the Krebs cycle and goes through a series of proteins, enzymes, and changes, goes through a biochemical pathway. Carbon dioxide leaves as waste product. ATP is created. NADH, FADH2 are formed. These are electron carriers. These electron carriers then go to the electron transport chain, where water, metabolic water, is produced, ATP is produced, and NAD and FAD are returned to the system. Okay? So we're going to go through that and look at those in a little more detail later, but today we're just going to concentrate on that glycolysis piece. Alright, so glycolysis is really just a sequence of proteins that occur in the cytoplasm, and these proteins that occur in the cytoplasm are part of a biochemical pathway. And if you remember when we were talking about biochemical pathways, that means that the product of one enzyme is the substrate of the next enzyme, and on and on and on. If you remember that video, so this enzyme's creating a product. That product becomes the substrate of the next enzyme, which creates a product, which is the substrate of the next enzyme, all the way till a final product is formed. The final product in this case is going to be pyruvate. Okay? And so how this works is there's 10 enzymes in solution in the cytoplasm. So kind of in a pathway in a linear form, per se linear, okay, that are going to take the glucose molecule. The first one's going to take the glucose molecule, and then utilizing some energy, some ATP, it's going to break that glucose molecule down. And it's going to go through the 10 enzymes until that 6-carbon glucose molecule is broken into 3-carbon pyruvate molecules, or 2 3-carbon pyruvate molecules. Okay? And so with that energy from the breaking of the bonds and the reforming and things like that, there's enough energy for phosphorylation to occur. And so you can phosphorylate ADP to form ATP. And when you do that, we call that substrate-level phosphorylation. So remember, the substrate is part of that biochemical pathway. So substrate enters the enzyme, product comes out, that's the substrate in the next one, on and on and on. Okay? And phosphorylation, again, is just adding a phosphate to this. So substrate-level phosphorylation. So in addition to making a little bit of ATP, there are some electrons that are free, okay? And with those hydrogen atoms, okay? And they get bound to NAD to form NADH. That's an electron carrier. You might remember from photosynthesis, the electron carrier for photosynthesis is NADPH. Okay? This one, there's no phosphate, so it's NADH. So we can look at this again, glycolysis kind of in a nutshell. You get glucose coming in, it gets phosphorylated by the expenditure of 2 ATP molecules. Two phosphates get attached, so you now have a 6-carbon sugar diphosphate. That 6-carbon sugar diphosphate is split, and you get 3-carbon sugar phosphate, 2 molecules of it. That 3-carbon sugar phosphate goes through an oxidation phase where electrons are removed to form NADH, and the energy from that is utilized to phosphorylate ADP to form ATP. And so you can see at the end of glycolysis, you get 4 ATP for that single glucose molecule. So you expend 2 ATP and you get 4.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Okay, so the video does a great job showing you the steps and everything, but I just want to correct it a little bit in that the end product, pyruvate, if in the presence of oxygen, okay, it will move into the mitochondria and get further processed and yield more ATP. If it's not in the presence of oxygen, it will form lactic acid. But this is only true for animals, the lactic acid part. Right? In plants and fungi and protozoans, if that pyruvate is not in the presence of oxygen, instead, in bacteria also, instead of becoming lactic acid, it becomes ethanol. Okay? And we'll look at this. This is how alcohol is created, okay? And so, or is formed. And that's from the not having oxygen to further break down the sugar. And most of you know this. If you add a little bit of yeast and a little bit of sugar and a little bit of water and close it off and not allow any oxygen, you will produce ethanol. You'll produce alcohol. That's through the processing of pyruvate into ethanol, not lactic acid. Okay, so just to correct that video a little bit. All right, so glycolysis itself yields very little ATP. I mean, you're getting a net gain of 2. So you had to spin 2 to get 2, and you know that's pretty poor when we calculated out how many molecules of ATP is needed to form 1 molecule of glucose through photosynthesis. We, you know, at a real over represented number, we'd say 60. The actual number that I kind of gave you, based on 9 molecules being utilized for per turn of the Calvin cycle. So we're talking about 54 molecules of ATP to form 1 molecule of glucose. So if you're taking that conversion, that's not very good. You're burning 54 to give you 2. It's not a very good process. But this is the only way that we currently know how organisms can convert food or convert glucose into energy when oxygen is absent. So if oxygen is present, there are other ways at which this can occur. And we'll talk about that from even the perspective of organisms that do not have mitochondria. When oxygen is present, they can further break down some of these molecules. And we'll talk about that later. The interesting thing, and probably one of the most evolutionarily important pieces, is that every single organism on the planet can carry out glycolysis. Everything. Bacteria, archaea, protozoans, fungi, plants, animals, all of them can do glycolysis. They can break glucose down via that 10 enzyme step to create a little bit of ATP. Okay? And you do this. You do this if you don't stretch and you just go run a race, or you just go run, up and down the halls or something like that. Eventually you'll feel a burn in the back of your leg. That's lactic acid. You just went through glycolysis without supplying oxygen, and you produced lactic acid, and that's the burn that you feel. Okay? And plants will do that and create alcohol or ethanol, and that's that's their kind of waste product, and our waste product is that lactic acid for the non-presence of oxygen. Okay. So to further kind of keep going on that aspect of evolution, since all organisms can do it, and we know that archaea are probably 1.5 billion years old at least. That's a really old biochemical pathway, and it hasn't changed much. It's not very efficient, but it works well enough that organisms can stay alive in regions where there is no oxygen. Okay, so with that, we're going to talk about pyruvate processing, the Krebs cycle, and the electron transport system in the next couple lectures.